and some about the kinds of things that we're doing with eDNA technology at SERC related to fisheries. And I guess I can start with just a simple definition for those who aren't familiar with it. eDNA is short for environmental DNA. And that just represents any DNA that you might collect by taking a sample from the environment. And so if as you go walking around your day through your house, go hiking on a trail, whatever, you might be losing individual hairs, or you might be sloughing off your skin cells, which you're losing all the time. And whenever you do that, it's leaving behind bits of your DNA that someone could potentially come along and sample. But that's true for animals. It's true for fish swimming around in the water that might be sloughing off their eDNA in their slime, or when they poop, or when they're mating. If they're fish that spawn out into the water, their eggs and sperm out into the water, like the river herring that are in this picture here that I'll talk about some. And so you can take different kinds of environmental samples. I'll be talking mostly about samples of water, or one of the cases that I'll talk about today is considering the stomach contents of fish as an environmental sample that we use eDNA approaches to analyze. But you can even, people are even taking eDNA samples from the air, collecting DNA that's floating around in the air to try to see what animals might be present or plants might be present in an area, and shedding their DNA. So it's really a kind of a catch-all term that is any kind of DNA that you might be sampling from an environmental sample. So I lead the Fisheries Conservation Lab at SERC, and our goal as a research lab is really trying to restore and sustain fisheries and the ecosystems that are required to support them. And we do a variety of different kinds of work. We study river herring, which I'll talk some about today, which are migratory fish. We study shark and ray migrations. We study blue crabs, of course, because this is Chesapeake Bay. I'll talk some about striped bass today as well. And we're doing a lot of research studying oyster reefs and oyster restoration. And in each of these cases, we're using eDNA approaches for some of the kinds of information that we're interested in learning and then applying to management and conservation. This background slide here gives just some examples of the kinds of things that we're using this for that are management questions. If you think about how we manage fisheries, most of the traditional fisheries management strategies involve things like regulating fishing effort, which might be how many fishermen can be on a boat, or how many lines you can put out, or how many crab traps you can have, what days you can go out, things like that. Or managing the catch, which would be something like the restrictions that have been making a lot of news for striped bass this year, reducing the number of animals that you can catch and keep each day. So that's a lot of the traditional fisheries management. And there are eDNA approaches that get at some of the questions to support that. But some of the examples that I'm going to talk about today primarily are things that we're using to answer this question about, can we manage the habitat that fish use as a fishery management tool? Are there things we can do to improve the habitat that would help improve fish populations and make it so that we can catch more fish and have more fish out there doing the good things that they do in the environment all the time? And the two examples that I'm going to use today are about restoring spawning habitat for migratory fish in rivers that was blocked by dams that kept them from swimming upstream to historical spawning habitat, and what happens when you remove the dam. And then also the idea of managing habitat in the environment so that there are plenty of prey out there for things like striped bass to eat. So this figure here shows striped bass and some of the other things that live in places like Chesapeake Bay, many of which might be prey for striped bass. So those are the two examples. And I'm going to start with the first part of one of the fisheries stories, which is river herring, to sort of set the stage for how we're using environmental DNA approaches. Then I'll talk through some of the details of those approaches, talk about the striped bass story, and then towards the end, come back to this river herring story that I'm starting out with. All the work that I'm talking about today has involved a lot of different people. And in each section, I've named some of the key ones here. And I will admit up front in this talk that I am not a geneticist by any means. So I work with people who are really good genetics that do the fancy genetics part of this. And my role in these projects is to think about how different tools like environmental DNA can be applied in management questions. And then working together with geneticists to design the studies, collect the samples, and then interpret the data that we get from it. So for this river herring work, the genetics guru was Lewis Plow, who has been at the University of Maryland Center for Environmental Science, Horn Point Laboratory over in Cambridge, Maryland. Although he just in the last month or so has taken a new job on the West Coast working on West Coast oyster restoration. So I'm very sad to lose him as a local collaborator, but we've done a lot of great work together over the last 10 years. And then all the other folks on this lab have helped with the field collections for fisheries habitat in the Patapsco River. If you're not familiar with the Patapsco River, it's the river that goes right between Baltimore and Baltimore-Washington International Airport. It's home of Patapsco Valley State Park, a beautiful state park. And it historically was one of the most developed industrial rivers in the US, going back to the 1700s. And that because of that industrial infrastructure, over the period of several hundred years, there were multiple dams built that restricted fish from being able to access different parts of the river. And as I've mentioned, the fish that we're studying there in particular are river herring, which is a collective term for two species. One is the alewife and one is the blueback herring. These are fish that live most of their life in the ocean and then come into freshwater streams in the spring to spawn. The term for that is anadromous. They're similar to salmon and other striped bass, other kinds of species that live a lot of their life in the ocean but need fresh water to reproduce. On the Patapsco River, the dam removal in particular that we've been studying for the last almost 10 years was the removal of Bloody Dam that was taken out by Maryland Department of Natural Resources and American Rivers and a whole bunch of different partners in 2018 and 2019. The dam was built in 1907, and it was one of the world's first, if not, I think it was the first hydroelectric dam that had the turbines built into the dam structure itself, built inside the dam. And it worked for 20 some years before the pond behind it became completely silted in. And since 1938, it was basically sitting there. It's about 3 stories tall. And it was a pretty big, expensive project to remove the dam. But after it was removed, you can see that the dam was taken out. The picture on the right is from just about the same perspective, looking at what the river looked like the summer after it was removed. And the trees have grown up some now, but it looks largely like this now. So there's some rapids there, but it's a flowing river that potentially allows fish to migrate past it. And I should mention in the dam, before the dam was removed, there was something called a fish ladder that was built. So this is like a passageway of little rapids that fish were hopefully going to be able to use to get past the dam. And then some kinds of fish used it, but unfortunately not really the ones that it was designed for, the river herring and shad. that were migrating upstream in the spring. For whatever reason, they didn't like the design of it and weren't really using it. And so one of the justifications of removing the dam, though there were several, was to allow them to finally be able to move upstream past this point and access places they had historically reproduced. So I wanted to introduce you to river herring a little bit more. One of the real tricks with studying river herring is that each species comes in over a period of about 6 weeks in the spring, and it's incredibly episodic. And so most days you don't see the fish. I personally, I've been working there 10 years. I have not once seen a river herring on the Patapsco River. I promise they're there. This was taken right next to the Maryland Food Bank in the lower part of the Patapsco River when the blueback herring were there. And one of the reasons we figured out that we almost never see them, this day, one of my staff got lucky enough to see them. We almost never see them because they mostly come in the river to spawn at night and then either leave again or hang out in the deeper pools that are hard to see during the day. We've used a sonar system that can see both during night and day and count fish to figure that out. That's not really part of the story, but it's one of the reasons that you don't often actually see these fish out there in the river. When they come in the river to spawn, a lot of times you'll see them in groups. This picture is actually taken over on the Choptank River on the eastern shore. But when they're spawning, what we often see is this fish in the front typically will be a female. And then there's a group of males following her. And these fish are broadcast spawners. So she'll release her eggs and splash a little bit as the eggs come out. So she releases the eggs, and then all the males swoop in and splash too and spread their sperm trying to fertilize those eggs. So it makes splashes like this that you can see. And this was a day that I was lucky and the water was really clear on the Choptank River. And I got some great pictures and videos of the spawning activity happening. So one of the reasons that we care about river herring, and we've been studying them, and we care about them in relation to removing dams, is their populations are maybe only 10% or less of historical populations. And that's due to two things. One is loss of their spawning habitat because of obstructions like dams or culverts or other things that block their migration. But also because after large refrigerated ocean going fishing fleets were developed and built after World War II, there were huge catches of these fish all up and down the east coast of the US. Initially, some US fleets, then some really large foreign fleets from Russia and Japan and other places. And then it kind of switched back mostly to US fleets, but you can see that this graph shows the catch of fish over time, over about a 50-year period. You can see that number goes down really low, and that corresponded to a really dramatic population decline. And then there was further decline in the 1990s, and we're not entirely sure what drove that decline, but the populations are probably somewhere around 10% of what they were historically. So there's a lot of conservation interest in bringing these fish back. And we want to bring them back for a variety of different reasons. And these are some of the management reasons. First, these are prey species or forage species. They're prey for lots of different things. So striped bass eat them, river otters eat them, bald eagles and ospreys and things eat them. So they're prey for lots of different fish in the bay, in the freshwater streams where they're spawning, and out in the ocean. So they're a really critical group of species that are moving from freshwater to saltwater every year. They can even be really important providers of nutrients to riparian forests along streams. So the picture here on the top right was after a flood on the Choptank River, where you can see a bunch of river herring, and there's also a hickory shad and a couple of white perch and things in there. These fish were stranded up in the forest, providing nutrients to animals and plants and helping to drive really productive forests along our tributaries here in Maryland. Then the bottom right shows the capture of a single large net on the Susquehanna River just to give you a sense for how many fish could be captured in fisheries in the rivers, before Conowingo Dam was built, for example. But the story is not all bad about dams in the Chesapeake. One of the most successful parts of the Chesapeake Bay program over the last 10 years, and really the last 30 years, has been reopening of a lot of rivers and streams throughout the Chesapeake watershed for migratory fish, and also for brook trout that are cold water fish that live mostly up in the mountains. But there's been really great success opening up a lot of rivers, and the Patapsco River is one of those more recent examples. So, so some of the questions that we had here for our research, and I'm going to kind of start going back into environmental DNA. We wanted to know for this dam removal on the Patapsco River, how many fish are coming in. We wanted to know if they were using the habitat downstream of the dam, both before and after it was removed. And then we wanted to know upstream of that fish ladder that I said they might have been able to use to access the upstream areas. Could they actually get past the dam before it was removed? And then once it was finally removed, did they move past it? We traditionally catch fish using nets or traps or other things. And I'll show some data from that as I get towards the end of the story. But that can be effective and time consuming. And as I mentioned, the fish were mostly moving around and accessible at night. So also might have to be at really painful times to regularly do research. The staff cost can be prohibitive, especially if you want to sample large scales. So my research group was working in the Patapsco River, but we also wanted to know across the entire Chesapeake Bay watershed, which is very large, how river herring were using habitats. And so trying to go around to hundreds of sites all around the watershed in a period of a few weeks in the spring is almost impossible unless you have a really large team of people. So if you can sample DNA from the environment or collect eDNA, if all you're doing is going out and taking a water sample in a bottle, that can be done really quickly and efficiently. You can store it and process it later in the lab. And at least depending on what you're doing, it can be a really cost-effective method because you can run, once you have the samples, which you can collect quickly, you can run a lot of them really quickly in a laboratory as well. But some of the times that it's less cost efficient is, if you want to know want to go out once and take a few samples somewhere to see if a fish is living in a spot, the fastest way to do it, if it's a common fish, might just be to go catch it in a net. The DNA piece could be expensive, but in general, it represents a potential cost savings. So I'm going to talk a little bit about eDNA, what the sampling we do looks like from my perspective as the fisheries scientist and not the geneticist. And I'll talk about some of the genetics as well. So this is my team going out on the Patapsco River to collect both a water sample for eDNA, and then the net she's carrying is for catching fish eggs and larvae that we call ichthyoplankton, basically fish plankton, fish that are floating around in the water. And this is at Daniels Dam, which is the last remaining large dam on the main stem of the Patapsco River. But basically what this looked like is going out into the field and scooping up some water in a bottle, or in this case, a plastic bag. They need to be sterile so that you're not like transferring DNA from one sampling site to the next, or you're not mixing in a bunch of human data or human DNA, or other things. So environmental DNA approaches are extremely sensitive to small amounts of DNA, and so we do things in as sterile a way as possible. This is a postdoctoral, well, at the time she was a graduate student at Duke University who was working with me on some projects, who's taking that water sample, putting it in these cups, and then filtering it through a really fine filter that captures the DNA and particles that might have DNA on it. on the filter and then passing the water through. So you go from having a sample of water, in this case, to having all of your DNA collected on a little filter. And then the part that I'm not going to talk in super detail about today is the part where the magic happens. You take that piece of filter paper that has DNA on it, run some chemicals through it that extract the DNA, that pull the DNA off of it. And then there's a couple different approaches that I'll talk about for how you identify or work with that DNA on there to identify what might have been in your sample. And then once you've done that, you send your extracted DNA from your sample off to a laboratory somewhere that has a machine called a sequencer that actually looks at the DNA and tells you every single little A or G or C or T for the nucleotides that or the chemicals that make up your DNA, and tells you this string of DNA. So when we go to a water sample and want to know what's in it, we get back data that look like this. That's great. There's a lot of information in there, but it's also pretty useless if you don't have anything to compare that to. And so the way that you find out what this corresponds to is you have to have a reference database or a reference library or something that we call, a lot of times this approach is called genetic barcoding. It's like the idea of having a barcode that you can read to tell what a species is. So we call it a barcode library. So does anyone, imagine nobody can guess what this particular species is, but it lives in Chesapeake Bay. This is, if this comes up here for me, this is a white perch. So this is a DNA sequence that we could get from a white perch. So how do we do the library? So this is one of the other big projects that we've been working on in my lab over the last 10 years or so. You need to have a reference library, and the way to get that reference library is to sample and sequence the DNA of all of the species that live in your area. And for a lot of things, you can do this using museum samples. So you might go to somewhere like the Natural History Museum for Maryland and sample maybe bird skins that have been preserved, or mammal skins that have been preserved, or plant tissue that's been preserved. Great. Unfortunately, most fish and invertebrates that live in the bay, so fish at the top, invertebrates at the bottom here, that live in the bay are preserved in formalin, which is a type of formaldehyde, and it degrades DNA very quickly. So they have essentially no useful DNA in them whatsoever if you go to museum collections. So what we've been doing at SERC and working with all of the different researchers and partners in the Chesapeake region who go out and regularly sample fish and crabs and other things, is to collect new specimens, 3 to 5 per species usually. We take their photo, as you can see here. Then we take a piece of muscle and extract the DNA from that and sequence it to get the genetic sequence. Then we also usually take another piece of muscle tissue and put that in a deep freezer, like frozen tissue bank that we have at Smithsonian. And then we also take the physical specimen, like what you're looking at the picture of here, and preserve that longer term in the collections at Smithsonian's Natural History Museum. So we now have over 80% of all of the fish and the crustaceans like crabs and shrimp and things, the mollusks like snails and the squid here, clams, oysters, and there's about, so for fish, that's 80% or 85%, I think now, of about 300 species in Chesapeake Bay. And for the invertebrates, we've done about 500 of maybe 1,000 species in Chesapeake Bay. So we're still working on this. But we've done basically all of the really common species, and we're just working on the ones that are rare, continuing to add those to our database. So that gives us this really nice local barcode library so that when we take a water sample or some other kind of environmental DNA sample for Chesapeake Bay, we have a great reference library that we can compare our eDNA sequences to figure out what was in our eDNA sample. So I mentioned briefly that there's kind of two primary eDNA approaches that you can use. And the stories that I'm telling today use both of those approaches. So I'm going to talk first, as I talk about these approaches, I'll talk first about metabarcoding, which is what we're using to find out what striped bass eat, what their prey are. And for this approach, you have something called a primer. It's a chemical that helps a chemical reaction that you do to increase the amount of DNA from your sample so that there's enough DNA to work with. Those primers typically target larger groups of species. For example, there are some that are designed to look at a specific section of DNA, small section of DNA that's really good for telling apart fish species. Or there are some that are more, they're called universal primers that target DNA or target sections of DNA that might help you tell apart fish, but also lots of kinds of invertebrates and other things. And so part of the research process using eDNA is to understand what types of things you're looking for, and then deciding exactly what kind of chemical reaction you need with these different sort of primers that help you target the species you want to look at. The nice part about this metabarcoding approach is that it lets you look at a lot of species at once. So like, what are all the different fish that are in the Chesapeake Bay? If they're there, can I find them and figure out what they are? And then the data that you get back tell you, how many sequences of DNA you get. Like, you might take some samples and get back thousands or even millions of individual sequences of DNA from your sample. And this will tell you, 50% of them were striped bass, and 20% were white perch, and 10% were something else, down to the rare things. So it gives you that sort of community level proportions of different species that you might catch. And that's similar to taking a big net and pulling it through Chesapeake Bay. And you're going to have a lot of some species and very few of other species that are more rare. So it's really kind of like taking a broad approach to look at all the different things that are there. The other group of approaches is more of a quantitative approach. So we can call it eDNA quantification, or what the term that's typically used for it is quantitative PCR. So PCR stands for polymerase chain reaction. It's just a set of chemical reactions that takes place to increase the amount of DNA that you have from your sample. So you're copying DNA for each chemical reaction, and doubling the amount of DNA that you have. And then if you do, that reaction in a machine multiple times, like 30 or 40 or 50 times, doubling the amount of DNA each time, your amount of DNA that in your sample is increasing. And if you set a threshold for a certain level of DNA, like whenever you have 10,000 copies of DNA in your sample as it goes through this whole series of reactions, for samples that start with a lot of DNA, they might cross that threshold after only a couple of reactions, a couple of sort of cycles of this chemical reaction. So you have a lot of DNA. But if you have to double the DNA with reactions a whole bunch of times before you get to 10,000 copies, then you know that you have very little of that species DNA in your sample. So it's kind of a nice approach, and it works really well if you can target a particular species. So in this case, you design these chemical primers for the specific species that you want to study. And so even though your water sample might have DNA from lots of different things in there, you're only looking at the DNA for that one species. And then you can find out, is it a little bit or a lot? Yeah, and so it allows you to sort of estimate the number of sequences that might be in your sample. So we use both of these approaches. And I'll talk first about identifying prey species that indicate habitat quality. This is the meta barcoding approach. The geneticist I work with for this is at Smithsonian. Her name's Katrina Lohan. She has the office next to mine, and we're working on a number of projects together. And for this project, so this is kind of a broader Chesapeake Bay management question than the dam removal question that I'll get back to in a little bit. For this, we work with the Chesapeake Bay program and scientists from around the bay, trying to identify what the prey items are that are most important for some of our key fish species like striped bass, but also other species that live in Chesapeake Bay. And we do that by looking at what they're eating and then trying to make sure that there's enough of what they like to eat out there in the environment. And if there's not, thinking about what can managers do to help improve habitat for those prey species so that there's more prey and then hopefully more striped bass, for example. So that was kind of the approach. And as the group working on this in a workshop got together, they called it the forage workshop, and foraged is just a fancy scientific term for prey species. They looked at what data were available already for Chesapeake Bay. And the best data on what fish eat and what striped bass eat and other species comes from a survey that Virginia Institute of Marine Science runs called the CHESMAP survey. And it's the Chesapeake, oh, I can't even remember the full acronym, Multi-Species Monitoring and Assessment Program. And so the map on the right here shows the number of striped bass that they've caught in their survey over a number of years. And everywhere there's a dot is a place they've taken a sample. And the darker the dot is, the more fish they caught in that particular sample. So you can see Maryland, the upper half of the bay here has a whole bunch of striped bass that they catch in their sample. And then they take a subset of the striped bass that they catch every time and all the other things that they catch, dissect out their stomachs, look at what's inside them, and do their best to identify it. And so that's kind of the traditional old school way of doing it. It works. It takes a lot of work, but it works. And they came up with these species list for several predators, including striped bass, some of the key prey items. So the ones in column A here on the left were the most important prey items, things like bay anchovy, polychaetes, which are little worms, mysids, which are little shrimp-like crustaceans, and various other things. Some interesting ones, some clams on here, mantis shrimp, which are kind of fun and crazy. But those were sort of the most important predators. The center column, column B, were a number of other important prey, but were less common. So things like Atlantic menhaden, Atlantic rock crab in the lower saltier part of the bay, blue crabs, a variety of other ones here. And then there was a group of species that the researchers who were in this workshop got together and said, we didn't see them very much in the stomachs of striped bass, but we know they're important and they should be on the list for one reason or another. So the American shad and river herrings that I've been talking about are on this list because they used to be abundant. They are no longer abundant, but they historically were probably one of the most important prey items. So they're on the list in the hopes that we can bring them back and they'll become important again. And then there's some on here, particularly mummichog and killiefishes and Atlantic silversides, which are small fish that are important prey fish, but they tend to be important along the edges of marshes in the tributaries. And if you'll notice on the map, all these samples are taken from the main stem part of Chesapeake Bay. It's a big boat, it samples in big water, and doesn't go into the tributaries. So the work that we were doing in my lab was to study striped bass from the little shallow tributaries, look at what they're eating, and add that to this database. And sometimes the things were easy to identify. So we did a little bit of the traditional method where you open up the stomach, look at things under a microscope and try to figure out what it is, and then count and weigh those things. So all the way on the left is an example of an easy one. It's a type of killifish. Not exactly sure which one, but it is a good enough prey item that you could identify it to species. The next couple in the middle are fun. So this is the mysid shrimp at the top, if you can see my cursor. The next one down and the little one in the middle here, this is a planktonic larvae of a horseshoe crab. And then at the bottom, this is the planktonic larvae of a barnacle. So these were all things that we found in stomachs of very small striped bass from the tributaries. Then you get to some other things. So the next one towards the right, you can tell from the legs that it's some sort of crustacean, but I have no idea what it is. And then you get to the right and there's this like partially digested stuff and no hope of identifying exactly what species that comes from. So you get a range of things when you do this kind of the old school way. So when they compiled those data, the data from the traditional methods in that forage or prey report, you get things like this where the height of the bar here indicates what percent of the stomach contents or gut contents of the fish by weight were different things. So some are obvious and easy, so bay anchovy, Atlantic menhaden. But then like for fish here, the third bar for fish is unidentified fish. It's a lot of fish material that couldn't tell what it was. That's a really great opportunity for genetic approaches because then you can identify those fish. The same thing happens for some of these other things. Here's a good amount of unidentified material, for example, in the green bars. So genetics can help us get at some of these rarer things or things that are digested enough that they're hard to identify. So for this project, we did a little bit of sampling on our own. Like this picture here, we did some sampling in the Road River, which is where SERC is located in Edgewater, catching striped bass that are all the way down from like an inch long up to a foot long or so. And then we obtained samples that are collected as part of both the Maryland Department of Natural Resources and also the Virginia Institute of Marine Science striped bass juvenile index surveys that they use to generate the annual year class data for management purposes. So we were able to get fish from those surveys to be able to take a bay-wide look at what small striped bass are eating in different places in the tributaries. When they come back to the lab, you can see some of them are bigger, like on this lunch tray at the top left, or small ones like the top right. We dissected out, we wanted to make the most use we could of these fish. So we dissected out their stomachs. We also did a bunch of stuff with parasites. So we pulled out their gills and their intestines, muscle tissue, took their stomachs for the stomach content studies. We took samples of liver and muscle, and then took out their otoliths or ear bones, which actually let us age how old the fish are. They have rings on them, sort of like tree rings, so you can age them. So I'm sparing you the gory methods here, but we took this, we emptied out the stomachs literally into a blender, either a small blender or a large blender, depending on the size of the fish, and ground them up into a slurry from which we extracted the DNA. And compared all of those genetic sequences of A's and G's and T's and C's to our barcode library from the Chesapeake Barcode Initiative. And this is sort of a very quick summary of what the results were like. We, for this study, looked at 188 stomachs. From those, we got back over 2 million individual sequences of things. Most of those were the same, so they represented common species that we had a whole bunch of individual DNA sequences from. but we were able to identify 1,247 different taxa. I'm going to call them species with very big air quotes. The scientific term we use is operational taxonomic units. So they're codes that are different, genetic codes that are different from each other that might represent different species or might also represent some genetic variation within a species. But we could pick out over 1,000 different things. I talked before about primers could be more general or more specific. These were very general primers that could look at everything from very tiny parasites up to very large fish. So we were looking at lots and lots of different stuff, which is why there are so many different potential taxa or species in this list. But mostly I was interested in the common ones. Katrina is a parasite and disease ecologist, so she was interested in a lot of the other tiny, rare, or unknown sequences that might be parasites or disease. So I won't talk about that today, but that's why we were looking at such broad different things. But this figure, which I'll walk you through, highlights some of the real value in this environmental DNA approach. So remember, we got these samples from sampling that's already happening for the most part. So we were able to get the fish from that. The dots on the map on the left are all the different sampling sites from those Maryland and Virginia surveys for juvenile striped bass, plus a few other collections that we did. And one of the things that we know about striped bass, but only from the Potomac River, is that the prey of small striped bass in their first year of life varies a lot depending on what the salinity of the water is, how salty the water is. So I've got the sites color-coded here by salinity, either essentially freshwater, very low salinity in the blue, some slightly brackish water in the aqua, and then saltier water in the red. Still not super salty, but 5 to 10 parts per thousand salinity, so a little bit saltier. And then in the middle, in the right, are the species that we got back from the environmental DNA data, grouped by those 3 salinities. So there's one column for each salinity grouping, from freshwater on the left towards saltier water on the right. And the thing that should jump out at you is that the color pattern is different. I'm not going to talk about all the different colors. You can see the sort of different groups of things listed over on the right. But there's a couple ones that I really want to highlight here that are interesting and that show sort of some clear patterns. So this bar on the left, these are the freshwater samples. And this green section in the middle, that's like half of the genetic sequences we got from small striped bass living in freshwater, are insects. So like half of the DNA that's in their stomach is insects. So insects are a really key prey item for them there. There's lots of other things. This red one are some small crustaceans called isopods. These are like roly-polies that live in the ocean or in freshwater, and some other ones. But that's kind of the key pattern here. And then you'll notice that this green bar very quickly gets small as you get to salty water, because there aren't very many insects that like having their larval or juvenile stages in saltwater. They're mostly freshwater. So that's a really clear pattern there. At the very top, these are amphipods. These are a small crustacean. Sometimes they're on beaches. We call them beach hoppers, but you see them really commonly throughout the bay. They're an important prey item, and they tend to like saltier water. So very few in the almost freshwater, and then they get pretty common in the saltwater. And then there's some that are really important in the middle salinity here. These are those mysid shrimp. So they like a little bit of salt, but not a ton of salt. And so there are areas where they're really abundant and important as well. So I just kind of wanted to highlight a few of the key ones here and show you that the salinity really does matter for what's in these stomachs. So how do we apply this for management? I showed this list before. This is an updated species list. So if we start in column A all the way to the left, the box at the top in blue These four most common things that were found in larger striped bass in the middle of Chesapeake Bay were also really common things that we found in the tributaries. So these are species that live both in the main part of the bay and the tributaries that showed up in both of our data sets. Same thing in the middle column. We saw in the DNA a lot of Atlantic menhaden and a lot of blue crabs. So confirming some of what was found, some of the prey that are found that are important in the main part of the bay are also important in the tributaries and all the way down to pretty low salinity. Then we found some really great data for this column C, and we're able to add a lot to it with our DNA data. Because if you remember, column C was just sort of based on expert opinion of things that people thought should be important, but were missing from the main bay diet data. And so we got a lot of evidence that the mummichog and killiefishes, these small marsh fishes, were really important. Atlantic silversides and other important prey fish were really important. And then we got a number of things that didn't make it onto that list. Insects were one that was really important. Soft clams, which are a clam, like if you eat steamer clams, that's what soft clams are. They used to be really abundant in Chesapeake Bay, but have declined also by something like 90%. So we found that they're important, even though their numbers are much smaller than they used to be. So there's a potential real management opportunity if we could restore soft clam populations that could bring back a really important prey item for striped bass and blue crabs and lots of other things. Grass shrimp live in marshes, we found those, and also striped blennies. And the interesting things about those is that those are, the striped blennies live on oyster reefs, and we found most of them in places where there's been large-scale oyster restoration. So that's a good sign that oyster restoration is bringing back prey for things like striped bass. So this is just a summary of the things that I was talking about in this section. We confirmed the importance of a lot of species that were already on the list, showing that they're also important in tributaries. We added some key species like insects and grass shrimp and soft clams, striped blenny. And then we were able to document really substantial variation by age of fish, which I didn't really talk about it, also by tributary and by salinity zone, which is the data that I showed here. And so the genetic barcoding, the eDNA approaches that we use to look at these stomachs provided species level identifications for many forage taxa. And before I stop talking about this meta barcoding sort of whole community analysis approach, this is an approach I don't have much data for our other studies to show yet, but I hope to soon. We're using this not on stomachs, but on water samples to look at the biodiversity that develops around restored oyster reefs. And we're about to start a new project using this approach to look at the species that are living in the Maryland offshore wind energy area off of Ocean City before, and then hopefully seeing how that potentially changes or not with the construction of offshore wind turbines. So this approach is becoming really common and usually involves water sampling, not grinding up stomachs, but those are the data that I had to show to illustrate this today. So let's go back into the Patapsco River and talk about river herring again in general. So I mentioned the challenge of looking at really large scales for these migratory fish that might only be around for a few weeks or even a few days each year. So we were using water sampling for these. Kind of our first level question was to actually build on some work that Ron Clauda, who I saw is on the call, had done a paper that he wrote several decades ago, looking at what river herring were still using in Chesapeake Bay and whether the population was small or large. Very few were large, but it was usually sort of large, medium, small, or kind of remnant, very small numbers in a particular tributary that was compiled based on sort of the best professional expertise of people. These river herring are in rivers in all different environments throughout the Chesapeake Bay watershed. These are just illustrations of how different some of the sites are. The center one at the top is the Northeast River, all the way up at the top of the bay. The one at the bottom left down here is the Chickahominy River in Virginia. There's some on the eastern shore here that are these coastal plain blackwater streams. So they're in lots of different places all throughout the watershed. And so we sampled all throughout the watershed with a lot of great volunteers who are pictured here, helping us to collect the samples. The map on the right here shows all the different places that we went over a 3-year period. The different colors just show what year or years we sampled each location. And when we did this, we collected 3 types of samples. One was if we could see the fish or throw a cast net to catch them, we would ID them based on the actual fish. You're looking at 2 of the fish that I got lucky enough to catch with my cast netting skills here. The one on the top is an alewife, and the one on the bottom is a blueback herring, but as you can tell, they look very similar. That didn't happen at very many sites at all. Then at each site, we collected a water sample, like in this bottom middle picture for our eDNA survey. And then the bottom right, she's using the ichthyoplankton net again to collect fish eggs and larvae to try to assess whether the fish are spawning and their eggs and larvae are free floating in the river. So 2 of the approaches here that I've been talking about, the eDNA approach, again, is DNA shed from the fish into the ambient water. We use this quantitative PCR or qPCR approach to amplify and quantify how many copies of DNA there are per liter of water that we sample. And that gives us a species level identification, which is great. Sorry, the bottom of that is wrong. It should say, well, it could, no, that's right. It could detect adults or eggs or larvae or juveniles. We don't actually know. We just know it's DNA from that particular species. The ichthyoplankton sampling that we were doing, we would put that net out in the water for 5 minutes to collect whatever fish eggs and larvae float into it, and then calculate the number of eggs per 100 cubic meters of water. So again, sort of how many eggs per amount of water is sampled. It unfortunately doesn't quite give us a species level ID because alewife eggs look basically the same as blueback herring eggs, look pretty much the same as hickory shad eggs, and sometimes gizzard shad eggs. So it gets us to the right group of species, but not necessarily the exact species we want to know. But it does tell us, which eDNA does not, if the fish are actually spawning there because there are eggs there and you know that they're reproducing. I'm just showing the DNA data, but wanted to sort of give you the sense that we're comparing, again, both the DNA approaches and the more traditional approaches to sampling. When we looked at the DNA data, each dot on the map is a sampling location, and the 2 species of river herring here, alewife and blueback herring, in each map. The black dots mean we took a sample there, but there was no DNA from that species at the dot at that site. And then the size of the circle indicates for each sample how many copies of DNA were in that 1 liter bottle of water, which could range from only a few copies up to 1 million copies or more. So a huge range that doesn't necessarily tell us exactly how many fish were there, but gives us a sense of, was it a little bit or a few fish or a lot of fish? And you can kind of glance at this and see that there's some patterns here. I won't talk too much about that, but you can see that the alewife, the dots are bigger and the red dots are more common on the eastern shore. And the blueback herring, the blue dots tend to be bigger and more common on the western shore of Chesapeake Bay. And there's some other patterns that we've looked into as well. But these are really the first maps that kind of update the information that Ron pulled together decades ago to give us a modern picture of what areas river herring are using during the spring spawning season throughout Chesapeake Bay. So that was a really great approach, and the eDNA approach allowed us to get to all of those sites in a relatively short amount of time. So, if we focus back on the Patapsco River, here's a map of the Patapsco River near Baltimore. If you can imagine yourself being a river herring that's lived in the ocean and is coming into the river to reproduce, to spawn, you would have to swim from the ocean. And we've found out in other studies that they actually spend most of the year kind of up in the Gulf of Maine, like north of Cape Cod. But they have to swim all the way down. Then they swim up the bay to the Patapsco River and are swimming from right to left in this picture, going upriver looking for a place to reproduce and mate. And so that you have that sort of first stretch from Baltimore to the Bloody Dam site, past Elkridge. And then I talked about Daniels Dam, and we'll talk about that some more. Ellicott City's there in the middle. I've already talked about Bloody Dam, but just to remind you of what that looked like. And we wanted to know, with the research that we were doing, whether removing Bloody Dam was restoring river herring spawning habitat use, and sort of reconnecting fish with historic spawning habitat. So we used a couple different methods for this, which should be a theme that you're used to by now. We wanted to know, are river herring detected at all above the dam site after it was removed? And for that, we used both eDNA and pit tagging, which I'm not going to talk too much about, but it's putting a tag that's like the microchip in your dog or cat into fish and trying to see if we can track individuals. And the Maryland Department of Natural Resources also used a fishing method that I have pictures of called electrofishing, where they were trying to catch adult fish. And then we wanted to know, are they actually spawning in that newly accessible habitat? And the only way we could tell that with our methods that we were using is by collecting the eggs, doing the ichthyoplankton sampling. So yeah, I've talked about the different methods here. Electrofishing is kind of this approach where you have a boat that puts electricity into the water that doesn't kill the fish if you do it properly, but it stuns them and makes them easy to catch. So you can go out and catch adult fish and see if they're using a particular part of the river. And because you get the actual fish in your hand, you can get species level information on those and estimate abundance. So we broke the river into 3 sections. There's sort of the downstream pink sites section at the bottom right. Then what we called the restored sites, or the restored reach of the river between where Bloody Dam used to be and Daniels Dam, a little bit further upstream. So those are the kind of yellow triangles or orange triangles. And then Daniels Dam also has a fish ladder on it that allows fish to get by potentially. So we sampled a few sites upstream of there as well. Those are the green squares, the sites above Daniels Dam, very cleverly named. And I'll show you data from some of the different sampling methods. So this is the Maryland Department of Natural Resources crew on their electrofishing boat that's putting electricity into the water. And then the guy standing up in the front of the boat has a long net on a long pole that he's going to use to scoop up the fish. And they sampled some sites that are downstream of the dam. Those are their ones on the left there. 591, 592, and 593 were all downstream of the dam. And they caught fish there both before the dam was taken out in the top panel and after it was removed in the bottom part. That's not surprising. These fish are coming in from the ocean and they should be there. They kind of have to be there to get anywhere else in the river. And in the first 3 years after the dam was removed, although 2020 was one of those years and they weren't allowed to sample that year, they caught 1 alewife and 1 blueback herring upstream of the dam. They've since caught a few more, but using this method, which took a lot of time and effort every week, once a week, spending a whole day on the river doing this, they caught 2 fish over the course of 2 years in the restored section. So it showed that the fish were getting there, but very little data to work with. With the data we got from eggs over the same time period, and we were able to do this during COVID, so it is 3 years of data after we were able to do this properly socially distanced. And so before the dam was removed, in that lower right-hand stretch of the river, we had a lot of eggs that we were catching at some of the sites in the lower part of the river, but we never caught any, and represented by not having any of the pink circles, we never caught any eggs upstream of either dam. So river herring and hickory shad, anything else that has an egg that looks exactly like that, we're not getting past Bloody Dam. before the dam removal over the course of 4 years of sampling. But in the 3 years after sampling, we also didn't catch any eggs. So we didn't find evidence from the eggs that fish were moving into that restored reach and spawning. But the story is different with DNA. Because DNA is incredibly sensitive to small amounts of, or eDNA sampling is incredibly sensitive to small amounts of DNA. So if you look at the picture on the left, before the dam was removed, we saw alewife DNA, the orange dots, or kind of, well, yellow dots, at several locations downstream of the dam, but never upstream of it. So we still didn't find any evidence that fish were going past Bloaty Dam before it was removed, but afterwards we did. We saw them at several locations all the way up to Daniels Dam, but not past Daniels Dam. They just got to that dam, but they were using the restored reach. So that was great news. For blueback herring, it was even better news. We saw them downstream before and after the removal, but in the panel on the right, you see that almost all the dots are blue, showing that they both got to Daniels Dam, but could actually use that fish ladder and go further up the river from that. So they were moving way up into the upper part of the river during the spawning season. So that's great. Still just the evidence that the fish are getting there, but not that they're reproducing. So over the last 3 years, we've increased the amount of samples that we've taken for the ichthyoplankton. And if you look at the right side of this figure, we've now also shown that we can catch river herring eggs in the restored reach up as far as Daniels Dam, at least. So we now know that they're also spawning in that restored reach, which is great news. So eDNA was a really great way for us to detect that they're getting there, and then adding on the plankton sampling for their eggs has helped us show that they're spawning there as well. So yeah, this combination of methods that we've used, eDNA has been a really powerful tool for detecting both the presence of river herring all throughout the watershed, and whether it's a few or a lot of herring. It's revealed some of these really large-scale habitat patterns, like preferential species use of one side of the bay versus the other. We've been able to show that using the eDNA that river herring are using habitat during the spawning season in every one of the rivers that we've sampled. Not necessarily every spot we've been to, but every river that we've sampled in, we've found some evidence from DNA that river herring are using them for spawning. And then we've, with eDNA, detected that habitat use in the restored reach after the dam removal, starting the very first year after the dam was removed, and every year since, and more reliably than the other traditional methods that we were using. We were much more likely to find them there than using the other methods. So it just shows kind of the power of eDNA for finding rare things if you're using this really targeted approach that targets a particular species. So I hope you've learned something. I've tried to explain things as much as possible, but I'm happy to stay as long as you want and answer questions. There are a lot of people to thank from this work, from the striped bass, especially Maryland Department of Natural Resources and Virginia Institute of Marine Science, for all of the collections of striped bass that they contributed. We've had technical assistance from a lot of people. I didn't list everyone, but from SERC, from my lab and Katrina Lohan's genetics lab, from Lewis Plow's lab at UMC's working on river herring for many years. And then for that Bay-wide survey, we had dozens of private landowners that allowed us to come onto their land to take samples. And that was a really key part of being able to do that study. And we've had funding from several different groups for this work. The striped bass work was funded by NOAA's Chesapeake Bay office. The river herring work has mostly been funded by the National Fish and Wildlife Foundation, and some of our recent work just in the last year also by American Rivers and NOAA. And then funding as well from the Smithsonian Institution. So I'm happy to answer questions that you have. And yeah, I hope you've enjoyed learning a bit about eDNA and some of the ways that we're using it for our fisheries research at SERC.
That was fascinating. Thank you so much. Why don't we come back and unshare? There are questions in the chat box that I'll have to go up here and see. Some of them were answered as we went through. Let me see here. Oh, let's see. Wonderful. Okay. Okay, so, well, let's see. Steve has, do you get ambiguous identification based on mitochondrial sequence? And could closely related species obscure the assigned ID? I don't know if that's more on the genetic side or not, but.
Yes, let's see, I'm trying to find, oh yeah.
Yeah.
So, yeah, I think what we're using for this is primarily the nuclear genes is what these are based on. This is one of the questions that I would normally kick over to Katrina for that.
Yeah.
But I think, you know, where there tend to be challenges in sort of in trying to identify things, there's several different challenges that we run into. And so the first one of them is that that if you have really closely related species like alewife and blueback herring, there can be very minimal differences based on the genetic code if you're looking at a relatively short piece of DNA like we typically do for genetic barcoding. So for for river herring, actually, if you use sort of the standard barcode for a region of DNA called CO1, which stands for cytochrome oxidase 1, which is a common one that's good for both fish and invertebrates. There are 655 base pairs in that, and a single difference between those two species in the region that targets. So you can use other parts of DNA where there might be more different, but for that particular kind of commonly used region for fish and invertebrates, or both vertebrates and invertebrates, when you have very small differences like that, you're less confident that it's 100% that species. You might be 97 or 8% sure, maybe even 99% sure, but getting to close to 100% is just harder because they're so similar. One of the other times that we run into issues is that some species have some genetic variation within that species. And so it might look like it could be multiple species that are very closely related, or it could be a single species that has more genetic variation within it in a particular section of DNA that you're looking at. And that can be really challenging to tell apart. Sometimes you find surprising things like, you actually find that what people thought was one species is actually 2 or 3 species that look almost the same, but they're actually genetically different enough that they are renamed as multiple species. So that happens sometimes. Or it can just be variation within a species that's, a little bit more than you would expect for most species in a section. So that's one of the times that there can be ambiguous data. And then, particularly when we do this meta barcoding approach where you're looking at the whole community, the data that you get back are only as good as the database that you're using. And so if you're using like global publicly available database, like GenBank is sort of the common one, there's a lot of things in there that people misidentified when they were actually looking at their specimen. So you might have one identical sequence that comes back as possibly multiple species because some people might have it correct and some people might have it wrong, and all of their information is in there. And so we sometimes get, strange things from Asia or Africa showing up in our data if we use the global database that's publicly available. But if we restrict that down to only our local database for only the species that happen here, then most of that goes away. So And a lot of times you can sort of trace, this fish was identified by someone, and you can trace it back to sometimes to the person that identified it. And there's usually not a sample to go back and look at, but sometimes there is. So people do things like, if someone has a question about one of our fish in our database, or an invertebrate, they can go to the Natural History Museum and look at it. So that's why we try to collect, all those different types of samples.
So there's
I know that's kind of a long-winded example, but or answer to that question. So there can be a lot of ambiguity, and it comes from multiple sources. So we sort of use our biodiversity experts at Smithsonian once we get the data back to help go through it and determine for each species that's in there, does that make sense or does that not make sense? And like one of the great examples that I mentioned and showed a picture of in our study is when we had these little striped bass that are an inch long or 2 inches long, they came back with horseshoe crab DNA. And you immediately think, how the heck is that possible? But when we actually looked in some of the stomachs, they had tiny little horseshoe crab larvae that they had eaten. So it totally makes sense. But on the face of it, looked crazy.
I love that. So Steve and Richard sort of have a question that are similar about the sampling. How often do you have to sample to get the consistent diversity in the region, and in order to determine the progressing status of a fish through a region?
Yeah, that's a great question. So there are a couple answers to it. One is that so there's how often do you have to sample, or how many samples do you have to take on the day that you go sampling is another thing too. So people take replicate samples. And in sort of with respect to that, sampling with eDNA is similar to sampling with a net. So if you go, to the Choptank River and pull a net a bunch of times, you're not going to catch the same thing in the net every time. You'll have a lot of the common stuff every time, but you'll get, the rare things might show up once in 10 net tows. And the same thing happens with DNA. The more samples you take, the better you do at describing the what the rare species are in the area, and sort of what the overall abundances are relative to each other. But to use the quantitative approach like we have for the river herring, I think it depends a little bit on your sampling question. But we've been doing some work to try to understand how the quantitative eDNA results that we get relate to how many fish are actually there at a site. And so we've been doing this in the Choptank River with a grad student at the Horn Point Lab. For that, I was using a high resolution sonar to count river herring every hour for 10 minutes for 3 months. And then once a day, she had like an automated water sampler that was out there taking water samples and duplicate water samples. So we had, every day for several months, we had counts of fish and an eDNA sample. And then she processed all of those data and found that she was able to, just based on the DNA, for that one location at least, reproduce the counts that we had if she just made a model based on a little bit of the data and then applied it to the whole data set, that she could get to within 5% or often less than like 2 or 3% different from what our estimated population counts were from the sonar method that we were using. So it's, we think that's probably sort of location specific. So you couldn't take that same relationship between the amount of DNA and number of fish and go to the Potomac River or the Susquehanna River, because it'll be different for each river. But if you want to track a population over time in a location, then you can do a pretty good job of doing that once you establish what that local relationship looks like. So we think it can work pretty well, but that's kind of the cutting edge of DNA approaches of trying to get at population size using these quantitative DNA approaches. So it looks promising. but it's not really been applied at large scales yet. But I think it can work with some caveats. But I should say that most of the population assessments that are done for fish like that also aren't great. So like some of the surveys for migratory fish, like river herring, sometimes some types of surveys like the electrofishing method that I talked about is done once a week, and during the daytime. So if you're working in a place where they're mostly active at night and they're really only there a few days a year, if you sample on Monday or Wednesday, you'll get completely different results that are hugely more different than what we estimate if you take a DNA sample every day. So some of the current monitoring methods we use make it really hard to tell population change as well.
Well, could you follow up with that? With the question is how long does the eDNA persist after a fish might no longer be in that area, and how that plays?
Yeah, that's a great question. That's also one of the huge questions that scientists have. And for the applications that we've been looking at, like I've been talking about today for the river herring, we estimate that it stays around at best for a day or two. So not very long. So it reflects what's been there for a relatively short amount of time. There's a lot of things that play into that, but when the water's warmer, you have microbial processes that happen faster and degrade DNA faster. So temperature affects the rate at which the DNA goes away. So the study that I was mentioning that the grad student did to try to estimate numbers of fish from eDNA in the Choptank River, she did several experiments through the course of the spring at different temperatures as the temperature warmed up in the spring to predict how long the DNA persisted. And it was really just a couple of days, a day or two. If you have a big pulse of fish that comes in and spawns and releases, a ton of DNA, it washes out of the river or degrades within a couple of days.
And we had a couple of questions regarding, do you find invasive species and or could it be used for endangered species use and for management?
Yes, absolutely. Those are great questions. So in the striped bass project, we put a couple of the paper that we wrote from that. We put a couple examples of that in the discussion part. So we picked up a number of invasive species that the striped bass were sampling by going around the bay and eating things. A lot of them we knew about. One of them was introduced to the East Coast. So people knew it was in the mid-Atlantic, but it had never been caught before in Chesapeake Bay and identified. But the striped bass got it in a bunch of different places. So it's definitely around in the Chesapeake, but no one's actually caught a physical specimen and identified it yet. And then we also had an example of a species that is commonly known further south that hadn't been recorded in the Chesapeake before. So it's local to the region, but is probably moving north with climate change or is just really rare in the bay. But the striped bass managed to eat some of those too. So we had the first record of it in Chesapeake Bay. Yeah. So definitely, and that's I'm talking about our study with striped bass stomachs, but the same thing happens with water samples. You can detect invasive species showing up in water samples.
And do you think that private landowners might get a little less open for y'all coming on the land because they might find endangered species and it would cause them some land issues?
Yeah, I mean, we didn't run into that. I mean, for that particular study, we were just looking at river herring and we weren't doing anything else with those water samples. So that topic didn't really come up. People were really interested some places we ended up talking to people for an hour. It slowed down our fast sampling approach. It was a lot of fun, talking to people and getting to know them, but people were really interested to know what was happening on their property with migratory fish. The thing that we actually ran into with that study is that there were people concerned about nutrients running off of their farms and polluting streams. So there were several people I had to swear up and down that I would not test for nutrients in the samples, and I did not.
Well, it seems like eDNA is changing the whole dynamics of research and design of studies. Are people having to go back to old studies and look at it again under the lens of eDNA?
Yeah, I mean, I think that's one of the things that's happening. It's kind of, it doesn't solve all problems, and it's got some of its own problems and challenges. Um, but it can be a really powerful sort of additional tool in lots of different ways. And so that's kind of how I view it. Is it's another, another way we can approach answering important questions that has some really nice features that other approaches don't have. So that's kind of how I think about it. And the striped bass example that I gave, that really allowed us to scale up. So that some of you may know Ed Hood, who's been at the Chesapeake Biological Lab probably since before I was born. I don't know, a long time. And so he was involved in sort of an early study of diets of small striped bass in the Potomac River. And we had samples from some similar, some of the same locations because those were also from the striped bass juvenile survey. Um, and so in a sense, we were repeating that study, but at much, much bigger scale than you would really be able to do, efficiently using the traditional approaches. And the other, that sort of the add-on benefit that we got from eDNA is I was talking about, these stomach content samples, is that this sort of ability to move past large amounts of things that are difficult to identify to the species level, to where we can get huge amounts of information on individual species that striped bass or other things are eating, all the way down to the species level, like what kind of amphipod it was, what kind of polychaete worm it was. Not just polychaetes are important, but exactly which species they're eating, which wasn't really possible before because they're hard to identify when they're not in the stomach and chewed up. So that's kind of a real, and kind of helps us expand the scale and also the precision of what we're doing.
Well, does anybody have any other questions for Dr. Ogburn?
Yeah, so there's one in here about like federally listed or endangered species. Yeah, so there's the same kind of species specific approach that we used for river herring. There are people using that for Atlantic sturgeon and short-nosed sturgeon. So those are some examples of endangered species in the Chesapeake that people use eDNA for. Um, and yeah, no, we haven't exactly overlaid our forage species in the presence of snakeheads, but that's, that looking at like the impacts of non-native predator fish on local communities of prey items is something else that could be done with funding, and we would love to do it. um, let's see. And then, yeah, there was another question in here about from Sherry about doing full genome sequencing or targeting specific regions. And that it was one that I kind of answered in the talk. So that everything that I presented was targeting specific regions, which was either the CO1, cytochrome oxidase 1 region, or a region of the 12S genome or the 12S gene. And it's a set of primers that are called MyFish that's kind of named after someone. But it targets fish. It's one of a couple sets of primers that people use specifically for fish. And we use those two because that's what is in our Chesapeake specific database. But one of our colleagues at the Natural History Museum is starting to do whole genome sequencing, which gives you huge amounts more data to work with to tell things apart. The reference libraries for that aren't as well established, but that's coming. It's an approach we want to use with our samples that we've archived in the museum. And We have a new project that I mentioned that's starting up that's going to be looking at potential offshore wind impacts and what species occur in the wind area throughout the year. And we may have an opportunity to start using whole genome approaches for those samples, in addition to the specific regions that we're looking at now.
Did we get them all?
I think we got just about all of them. Yeah, so this has been a question too, the Maryland darter. So not to my knowledge is that, and so we don't have Maryland darter in our DNA database either, because I'm trying to remember. My recollection is that all of the samples are preserved in a way that it's difficult to get DNA from them. So I don't think we have it in our database, but I'm not 100% sure that's true. But I think the samples were in formalin, sort of most of the museum samples at least. So it may or may not be very possible to do it, but it's definitely something people have been talking about.
And we recently had a presentation on the Maryland darters, so that you can go back and take a look at that as well. It's great. We've been
Great.
on a
Well,
fish kick. It's been
yeah.
good. And we're going to continue on next week when we learn about fish tongues and other oddities. So that's what maybe you want to come back and join us as a participant, a listener, to learn about that. You're more than welcome to, as are all of you, to come back. But thank you so much, Matt, for your time, your expertise, and all that you're doing to help us understand our natural world, the habit, and what we need to do to improve the habitat and ecosystems for fish in Maryland. We really appreciate all the work that you and your colleagues are doing out there in the field.
Yeah, thank you. Well, this has been really fun. I appreciate everyone listening. I know I tried to pack a lot of information in today, but it's been great to be able to share it with you and answer your questions. And yeah, it's been a lot of fun.
Yep, we're all smarter for it. And everybody stay well, stay curious, stay outside. And even the fishing people sometimes go out and don't catch any fish. So that was also. But now that we have eDNA, we can catch fish all of the time. It's a different type. So everybody stay well. We'll see you next week for Fish Tongues. Take care.